And, to me, it just didn't seem that it could all be about personal responsibility and it could all be about, willpower, which was kind of what the mantra was back then. And looking at our changing food environment, we started to kind of see, well, there's a lot more sugar in our environment than there was 30 years ago. And now there's a lot more obesity than there was 30 years ago. So perhaps that has something to do with it. So when we developed this animal model, we basically wanted to assess, whether or not binging, withdrawal, and craving, which are kind of like the triad of behaviors associated with addiction, would emerge in response to animals that are just binging or given daily access to large amounts of sugar.
Hmm.
And so we gave them a sugar solution. They always had water to drink too, so they were never forced to drink it for hydration. They always had food available, so they were never forced to consume it just for nutrients. And what we ended up seeing was that, in fact, the animals that had this extended access to the sugar solution would develop binging. They developed signs of tolerance. So, they would have larger binges throughout the day. They'd also show signs of withdrawal. And this was measured using measures of depression. We also used the elevated plus maze to assess anxiety. And we also had somatic indications that we were assessing, like, changes in blood pressure, changes in body temperature, and even just like ultrasonic vocalizations of distress, essentially,
Hmm.
that they can make to each other that we can pick up using different devices. And then lastly, the craving part that really, can be measured in animals, even though they can't tell us they're craving something. We can do different experiments to see how hard they're willing to work to
Hmm.
get access to a substance. And that's usually an indicator that they're craving. So we used operant conditioning to do that. And, essentially what happens is that, we're able to demonstrate these behaviors in animals that are having this excessive consumption of sugar, but not in animals that are eating regular rat chow or animals that are having sugar just occasionally.
Hmm. Yeah, and I guess one of the keys here is that, you said they always had access to plain water, they always had access to other food, so they can opt in to doing this. And even though they
Yes.
have that option and it's not necessary for them to hit their nutrient or water requirements, they often will choose to do this, and the binging becomes more frequent, it becomes bigger, and you can essentially measure things like a mouse sugar hangover.
Yeah, basically that's what we've been doing. And, also in people too, because there's been some scales that were developed and ways in which a lot of the work that we started off doing in our animal models could then be translated into humans. And so many of these things can now, and again, with the use of neuroimaging techniques, we can basically see this happening in humans as well. And I think one of the things that, for me is so exciting is that we're able to, capture a lot of what's happening in the brain because a lot of times behaviors, yes, they are indicative of what's going on in the brain, but to be able to really see these neurochemical changes happening in real time, because a lot of the techniques that we were using at the time of these initial studies were in vivo microdialysis techniques. So that basically allows us to cannulate the rat in the brain and extract extracellular fluid while the animal is awake and moving. So they're basically running around the cage tethered so that we can collect the sample from their brain, and they're eating, they're drinking, they're, engaging in whatever behavior they like. And so to be able to see the rat drinking sugar and then look over on the screen and see a spike in dopamine, I mean, it's really telling to see that
Hmm.
happen.
Hmm. Yeah, so basically, you see all these behavioral signs that the animal is in fact becoming dependent or engaged in compulsive behavior, and then you're saying basically what you've said so far is that you can see in the brain that the consumption of sugar by rodents is eliciting the same types of effects and the same parts of the brain that you would see in response to an addictive drug.
Yep, absolutely. And, when you say it like that, it kind of sounds like, oh yeah, that makes a lot of sense. But I have to tell you, when we first were doing these initial studies, like when I was a, early grad student and I would go to conferences and present some of our new stuff, it wasn't well received. And it was really just